Hey CW Bio, Mr. Kennedy here. Today we're back in chapter 11 of your book looking at Mendelian genetics. I'm going to take a little time here and teach you, well, the vocab and all the steps that you need to know on how to do monohybrid and dihybrid crosses using a Punnett Square. In order to be successful with these, there's a few vocabulary words you're going to need to know, and then you just need to practice the steps, and it'll be easy. So my challenge for you is to grab yourself a piece of paper, something to write with, get yourself a, drink and a comfy chair. Let's dig in. So first thing I'm going to do is I'm going to make a list of vocabulary words for you. We're going to start with the word monohybrid cross. Then, of course, we're going to do a dihybrid cross. Then we're going to talk about your genotype, your phenotype, and what it means to be homozygous, and what it means to be heterozygous. All right, so let's add the definitions here. When you do a monohybrid cross, you are crossing one contrasting trait between two individuals. So it's a cross of one contrasting trait. Okay, so maybe we're going to cross like a tall plant with a short plant. So I'll put that as my example. Tall crossed with short. Okay, next is the dihybrid cross. This is two contrasting traits at the same time. So a cross of two contrasting traits at the same time. So maybe now it's a tall plant with red flowers crossed with a short plant that has white flowers. So tall, red flowers, crossed with short, white flowers. Okay? Genotype. The genotype is your genetic makeup of the organism. Okay, so it's the genes that you have. So in terms of your genotype, if you were tall, you could have two alleles for tall that are like big T, or you could be big T, little t, and still be tall. If you were short, then you'd have to have like two little t's. So I'll put a semicolon there, and I'll put like little t for short. Okay? So those are your genotypes. Your phenotype is what you see in the mirror. So it's what is expressed. So you could say what you see, okay? Or you could say it is your expressed genotype, okay? So if I had a tall plant that was big T, well, your phenotype would be tall. Or if you were little t, you would be short. So I'll just write the words, tall or short. Homozygous. Homozygous means that the alleles for a particular trait you have are the same. So homozygous means you have two of the same alleles. That means you are big T, or little t, two of the same alleles in regards to being tall or short. Heterozygous is where your alleles are different, right? So I'll put alleles are different. So in that case, maybe you have one big T and one little t. That's what it means to be heterozygous, okay? All right, so you got to keep this vocab fresh in your mind. This is stuff that we're going to use as we do all of our Punnett Squares. What I'd like to do is to just walk you through Mendel's experiments as I teach you how to do these Punnett Squares. You should remember these from a previous video that we've done together. And I'll set it up like this. So I'll just put this line across the page, sort of separate the page, move this vocab out of the way. And let's start off with what Mendel did first. If you remember Mendel's experiments, he created pure breeding strains of 7 different pea plant varieties. So he had pure breeding tall plants, pure breeding short plants, pure breeding red flowering plants, pure breeding white flowering plants, and so on, so on, so on. Okay? So why don't we do our first cross like he did with a pure breeding tall plant crossed with a short plant? Okay? We're going to make the assumption that tall is always going to be dominant to short. So I'm going to write this out as a word problem because you need to get used to reading these things as word problems. It's how it's going to show up on your test. It's how it's in your book. So just get used to it because, yeah, like that's the way it is. So for our first cross, we're going to cross a pure breeding tall plant with a short plant. All right. How do we know what the offspring are going to look like? Well, we got to do a Punnett Square. Most genetics problems will set up the problem just like this. They'll say, cross this. And then they're going to tell you what they want after you do the cross. So let me finish this sentence with what I want you to produce for me. Okay? Cross a pure breeding tall plant with a short plant. Okay? Comma. Give the genotype and phenotype of the F1. Okay? So I'm using your vocab. The genotype would be the genes of the offspring, and the phenotype would be what they look like. Okay? And those will come out as ratios. F1 stands for filial 1, which basically means that first generation that are produced from these parents. So how do we set this all up? We're going to start by just going back to Mendel's rules of unit factors and simple dominance, recessiveness, and all of those sorts of things that you learned in a previous video, and we're going to apply them to this problem. Let's start with the rule of unit factors. Mendel said that for every trait that we possess, and we being in this case a plant, you have to have 2 alleles for that trait, one from mom, one from dad. So when we set up our problem, I'm going to make dashes like this to represent my parents. And I'm going to make 2 dashes on either side of an X. The X means cross. And these dashes are to represent the alleles for a trait that I'm crossing. Now, what trait am I crossing? Well, if I go back to this problem, I'm crossing a tall plant with a short plant. Okay? So that's one contrasting trait, tall and short. So I'm basically going to have to do a monohybrid cross here. What I need to know is what's the genotype for this tall plant and what's the genotype for the short plant so that I can put it on these blanks. Okay? Remember, you need 2 blanks because for every trait you have, being tall or short, you have 2 alleles for that trait, one from mom, one from dad. So the question says you are pure breeding tall. Pure breeding means that, well, this plant can only produce tall offspring and nothing else. Pure breeding means that this plant has been inbred to the point where, every generation that this plant produces comes out tall. Pure breeding is genetic code for the word homozygous. Okay? So if you're homozygous for tall, that means that we have to put 2 of the same alleles on these blanks. The question is, what do those alleles look like? Well, a moment ago I said we're going to assume that tall is dominant to short in all of our crosses. When we name our alleles, we use the first letter of the dominant trait to name the alleles as we put them on our blanks. So if tall is dominant to short and this tall plant is pure breeding, that means these blanks get 2 uppercase T's, capital T. Now, what about the short plant? What's the deal with that? Is it homozygous? Is it heterozygous? I don't know. Well, actually, you do know. If tall is dominant to short, the only way you could ever get a short plant is to have 2 lowercase t's as your genotype. Anytime you have an uppercase T, the offspring is going to be tall. Here's what I mean. So if I have big T, well, that offspring is tall because tall is dominant. If I have big T, little t, that offspring is still tall because according to Mendel's rule of dominance and recessiveness, if you have an uppercase letter like that big T, which represents tall, it's going to hide the lowercase letter, the little t, when it's expressed. So a heterozygote individual that's big T, little t, they're going to look tall. The only way you could be short is if you have two little t's. All right? So just by saying short plant, I should know that that individual is homozygous, because that's the only way you can have that happen. I don't have to write homozygous short, and I don't have to write pure breeding short, because there's no other way to be short unless you have a homozygous condition. So there you go. Okay? Now the next step in all of this is to determine what your sex cells will hold. So as a result of meiosis, each of these parents is going to produce two sex cells. So we'll just make circles underneath. And the reason I know that they're going to produce two sex cells is because if we imagine this as a single diploid cell, I'm putting dots around it to make you imagine that it's a cell, we need to cut the amount of genetic material inside that cell by half so that sexual reproduction can occur. That's what meiosis is all about, okay? When meiosis occurs, we're going to get two sex cells, each one with a big T in it, just like that from the first parent, and two sex cells, each one with a little t from the other parent. Now, we're going to use a device called a Punnett Square to predict the outcome of this cross. R.C. Punnett created this mathematical device that we use for monohybrid crosses that simply has 4 boxes to it, and we set it up like this, okay? Now, since this is something of a cross multiplication table, it really doesn't matter which parent we put on the, which side, because it's going to come out the same. 1 times 2 is still 2, and 2 times 1 is still 2. Doesn't matter what order you do it in it, it's the same. So we're going to take, just for, the sake of convention, the first parent, and I'm going to put the first parent on the side, and then the second parent on the top. Now, I put 1 allele for each row and each column, just like you see there. Now that that's done, I'm going to take an allele from the side, and I'm going to match it with an allele from the top to fill in this box. So this box gets big T, little t. Now, if you're paying attention and you're smart, you should realize that all the boxes are actually going to get the same thing, right? Because another allele from the side, another allele from the top, big T, little t, big T, little t, big T, little t, and so on, okay? So how do I come up with genotype and phenotype? Well, it works like this. I'm going to write genotype right here. Basically, it's kind of like a matching game. I start with this box. This is box number 1 of my Punnett Square. Then it goes 2, 3, and 4. And I'm going to ask myself, okay, how many boxes look like box number 1? All of them. So I write it like this. 4 over 4 is big T, little t. That's what's in them. Okay. Now, if you turn that in as a math problem to your math teacher, would your math teacher be okay with that? 4 over 4? No. They'd go, reduce your fraction. So if we reduce this, 4 out of 4 is basically 1 or 100% big T, little t. This is your genotype. Easy. Okay? Phenotype. It works the same way, but realistically, we have 2 options for our phenotype here. We could be tall like the first parent, or we could be short like the other parent. So I'm going to write them out. I'm going to say basically how many boxes are going to be big T tall versus how many boxes are going to be little t short. And as I look at all of my boxes have a big T and a little t in it. If you remember what I told you before, okay, if you have big T, little t, you're heterozygous. The capital letter is the one that's expressed. So you're going to get 4 out of 4 that actually have a big T in them, making them tall. Do you have any that are little t, little t? So 0 over 4 little t. Okay, we reduce this and get 100% big T as our phenotype. Easy, right? No problem. All right, so let's go on to the next practice problem. So we're going to take what we did on that page and we're going to expand this a little bit. So we're going to take and we're going to cross the F1 in that scenario. So for problem number 2, I'm going to cross 2 heterozygous tall plants. Okay? And still give the geno and phenotype, right? And in this case, this would be, we'll still call it the F1, all right? For the F1. How do we set it up? Well, we got to start with our parents, okay? So for every characteristic we have 2 alleles that give us that characteristic, right? You're told that you have 2 heterozygous tall plants. Go back to your definitions. If you're heterozygous tall, you are big T, little t, crossed with big T, little t. I'm going to go a little faster, so you should be getting it now, all right? They go through meiosis and produce their sex cells. So we get one with the big T, one with the little t, one with the big T, and one with the little t, just like that, okay? We're going to do our Punnett Square. And we're going to put 1 parent on each side. Okay, fill in the boxes. Big T, big T, big T, little t, big T, little t, little t, little t. There you go. Okay? Genotype and phenotype. Remember the matching game. So here's your genotype. Okay, how many boxes look like box 1? 1 out of 4 has big T in it. How many boxes look like box 2? Okay, 2 out of the 4. So we're going to make a ratio here. 2 out of the 4 are big T, little t. And you got 1 box left over. 1 out of the 4, little t, little t. Now, your math teachers will tell you that this is a ratio. They'll also tell you it can be reduced. In a very simplistic sense, I'm going to skip a whole bunch of steps and not do this right according to a math teacher. And I'm just going to multiply this whole thing by 4 over 1 again and again and again to cancel out all these 4s. Okay? So your final ratio of your genotype would be 1 big T to 2 big T, little t to 1 little t. Something else you need to know. In this ratio, those 1s and 2s actually still stand for percentages. So if you have a 1, that's like saying 25%, or again, 1/4 of your boxes, right, are big T, little t. If you have a 2, that's 50%, right, of your boxes are going to be heterozygous. And then you have, again, 25% at the end. Okay? Let's talk phenotype. Phenotype is what you see. I'll go ahead and write it out, phenotype. So, again, you can be tall or short. So we're looking for big T's or little t's, okay? And we can just use the ratio from above, and we can see that we've got 25% or 1/4 are going to be big T. Another 2/4 or 50% are going to be big T, little t. So you can just add those up. 1 plus 2 is 3. They all have big T's. So basically 3 over 4 are going to be like big T. That leaves you with 1 that's little t. So 1 over 4. Again, this is a ratio. We can reduce that, using that cheater system that Mr. Kennedy showed you, where you just cancel out your 4s. So your phenotype is 3 big T to 1 little t. And again, those numbers mean something. So the 3 actually means, right, 75%, and the 1 still stands for 25%. On your test, you could be asked for the ratio. You could be asked for the percentage. Doesn't matter, okay? You should be able to crank out either one at this point, okay? One final thing that I want to mention before I do the last 2 practice problems for you is that when we do a cross like this, there's no guarantee that your first offspring is going to be what you see in box 1. The idea is that every time this cross happens, these odds replay. It's like flipping a coin or rolling dice. When I flip a quarter, if I get heads on my first toss, there's no guarantee I'm going to get tails on my second one because, well, you already got a head, right? Like, I could flip that coin 100 times, and the idea is that after 100 coin flips, then I should be roughly 50-50, because that's the odds of flipping heads versus tails on a two-sided coin. So in this case, right, if this pair of plants only had one offspring, there's no guarantee that that first offspring is going to be tall. That first offspring could actually be short, and then you get a whole bunch of tall ones. I don't know. It's all in the game of probability and playing the odds. So here's two other crosses that you should be able to do. Okay, so we've crossed two pure breeding individuals. We've crossed two heterozygous individuals. So what if we cross a heterozygous tall plant with a short plant? What happens then? We're just going to assume you're going to do genotype and phenotype. Okay, like always. What does this look like? Well, we'll just go through the process again. What do my parents look like? Heterozygous tall, big T, little t. Short plant, the only way you can be short is little t, little t. Can't be short any other way. Okay, we do our Punnett Square. I'm going to go quicker because you should be getting it by now. And one parent on each side, right? Assuming we did meiosis here. And we've got these sex cells, right? Then this is what our Punnett Square will look like. Okay? We fill in the boxes. Big T, little t, big T, little t, and then a whole bunch of little t's down here. Okay? Now, genotype and phenotype. Don't want you to get confused, so I'll put a line right here to separate that out. Genotype. Okay? So here, how many boxes look like the first box? You should have 2/4 with a big T, little t, and that leaves you with 2/4 that have little t, and again, we can reduce using the 4, right? But that leaves me with a ratio of 2 to 2, which can still be reduced. So basically, this becomes a 1 big T to 1 big T to 1 little t ratio. This is your answer. Okay, and in this case, when it's like this, those ones aren't 25% anymore. Now they're 50%. This is literally saying you have a 50-50 chance of getting a heterozygote tall or a short plant. 50-50 chance. So then when we go to phenotype, okay, same idea. Can you be tall or you can be short? So if we use the ratio from above, how many have a big T? 2/4 have a big T in it. And how many have 2 little t's? 2/4 have a little t. We reduce that all the way down the way we did from above, and we'll end up with a 1 big T to 1 little t phenotypic ratio. That's your answer. Okay? 1 big T, 1 little t phenotypic ratio. So again, you could have a 50-50 chance of being tall or short. Okay? Now, the fourth and final one that we're going to try is we're going to try crossing a heterozygous tall plant with a homozygous tall plant. Okay? So I guess all the offspring are going to be tall. But what's the genotype and what's the phenotype? Okay? Let's look at that. Okay? Heterozygous tall plant with a homozygous tall plant. So we'll lay out the parents. Big T, little t, crossed with big T, big T. Okay? They do meiosis. And we do our Punnett Square. Okay? And one parent on, oops, forget that. Oh no. Well, okay. We'll go like that this time. Okay? All right, and then we fill in our boxes. Big T, big T, little t, and big T, big T, big T, little t. There you go. Okay, move this up so you can see it. And that's what our Punnett square looks like. Now we'll do genotype and phenotype. Geno and pheno. Okay, so matching boxes. How many boxes have big T in it? You have 2 out of 4 with big T, and 2 out of 4 that have big T, little t. We reduce that all the way down, and we're going to get that ratio like we did from above, that is 1 big T to 1 big T, little t. That's your answer. Okay? Basically 50-50 chance, right, of being homozygous tall or heterozygous tall. As far as phenotype is concerned, we can be tall or we can be short, but remember, the only way you can be short is if you have 2 little t's. Do you see anybody up there that has 2 little t's? I don't. Okay, so basically it's 4/4 that have big T's, right? 0 out of 4 have little t, so it's 100% big T or tall. That's your answer. Now, here's the beautiful part about what Mr. Kennedy has just taught you, ladies and gents. I've gone through really the only 4 possible varieties of monohybrid crosses that are out there. If you can master these crosses, if you can remember these ratios, if you could just remember the steps in the vocab, you can solve any monohybrid cross that I could throw at you on a quiz or a test. The only thing that could make things tricky for you is I might change the letters. So instead of tall versus short, it could be red flowers, white flowers. It could be yellow peas, green peas. Like, that's literally the only thing we can do to make it wonky for you, okay? So make sure you study this. Now, I'm going to do one other scenario for you. Flip this over. We're going to look at a dihybrid cross. And to be honest with you, like, it's kind of the worst of the worst in terms of Punnett squares. So, bear with me. We're going to cross 2 individuals that are heterozygous for 2 traits. So we'll just make it really ugly. And if you could do this one, then you can do them all. All right. So we're going to cross a heterozygous how about I spell that right? Heterozygous red flowering plant. That's tall with another tall red flowering heterozygous plant. Okay, so what this amounts to is you've got 2 heterozygous red flowering plants, and you want to predict what their offspring are going to look like. Well, since we're talking about red flowers and height, that's 2 characteristics at the same time. So now instead of 2 blanks per parent, you need 4. So it's 1, 2, 3, 4 crossed with 1, 2, 3, 4. Okay? You're told they're heterozygous, so that means you need some combination of tall and lowercase alleles here. We're going to assume that tall is still dominant to short, so that means there's going to be some big T's and little t's. And just for the sake of ease, we're going to assume red is dominant to white. So if you are heterozygous red flower tall, then that means you're going to be like, I always like writing the tall first, so and it really doesn't matter which one you do. You could do the height first and then the flower color second, or vice versa. It doesn't matter. Come out the same way. But if you're heterozygous tall, it's big T, little t. If you're heterozygous red flowers, you're big R, little r, like that. Both parents are the same. Okay, now meiosis has to occur. And remember, if we're starting off with a cell that has 4 alleles in it, then each of the cells that meiosis produces has to have 2 cells in it. If you remember your math classes, you probably learned this little thing called FOIL or JAWS in math. That's how we determine the combination of alleles for the sex cells in a dihybrid cross. So I'm going to take the first letter of the first trait, and I'm going to combine it with the first letter of the second trait. And that's going to give me a sex cell that has a big T and a big R in it. Then I'm going to take the first letter of the first trait to the second letter of the second trait, and now I'm going to get another sex cell This one's going to have a big T and a little r. I repeat the process like this on the inside. So I get a sextile that has a little t and a big R and a little t and a little r. Both parents are the same. Okay? Big T, big R, big T, little r, little t, big R, little t, little r. All right, that's how it works for meiosis. Now the Punnett Square. We're dealing with double the genetic material, so yeah, the Punnett Square's got to be double the size. You ready for this? Okay, it's big. All right, so you're going to need a Punnett Square that has 16 boxes in it. And these boxes are numbered like a typewriter type. So that's box 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. You get the idea. 14, 15, 16 boxes. We still put our parents on the sides and tops with the same sort of division for rows and columns. So big T, big R, big T, little r, little t, big R, little t, little r, big T, big R, big T, little r, little t, big R, little t, little r. There you go. Okay, are you sitting down? I hope so, because now we got to fill the boxes in. Like letters together, capital letters go first. Here we go. Big T, big R, goes in the first box. Next box is going to get a big T, big R, little r. Watch this. Box 2 and box 5 get the same thing. Big T, big R, little r. Okay? Then I've got to do a big T, big R, and this guy is going to get the same thing. Big T, big R. And then I've got a big T, big R, little r, and I'm just going to fill those in all the way down the middle because that's where they go. You'll learn this too with practice. And then I've got a big T, little r there in the middle. Okay. I'm on box 8, and I'm going to do a big T, little r, big T, little r in box 14, and little t, big R for box 11. Little t, big R, little r here. And the last one is all little letters. Whew, that's a lot of stuff. Wait till you see the genotype and phenotype. All right, matching game time. Better leave yourself some room for genotype, okay, because it's a doozy. So for the sake of argument, we're going to skip like the, fraction reduction thing and just assume that, you're going to do that. But for now, here's how I'm going to lay this out. We're going to start with box 1, and I'm just basically going to ask you how many boxes look like that out of the 16. And the answer should be 1 out of the 16, and it's going to be big T, big R, and I'm going to cross that off so that I know that I'm done with it. Then I'm going to go and make my little colon there so I can continue my ratio. I go to box 2. How many boxes look like that? Remember how I filled them in? So 2 and 5 are the same. So you write 2, big T, big R, little r, and I'm going to cross these out because I'm done with them. Okay? Then I'm going to go on to box 3. How many boxes look like that? Remember how I filled them in? 3 and 9. So 2, big T, big R. I cross those off. Okay, I'm going to keep my ratio going. It's getting scary. I go to box 4. How many look like that? Remember how I filled it in? 1, 2, 3, 4. So 4, big T, big R, little r. And I've crossed them out, so I know I'm done. I pick up box 6. So I've got one of those because none of the others match. Big T, little r. Done deal. And then I go to box 8. Remember how I filled it in? 8 and 14. So 2, big T, little r. Okay. And then I pick up box 11. 1, little t, big R. Done there. 2, little t, big R, little r's for box 12 and 15. And then I've got one left, little t, little r, little r. Man, that's your genotype. 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1. Whew. Yeah, that's your genotype. You can imagine under each of those is the number 16, and yeah, you'd have to reduce. But there you go, okay? We're not even going to worry about percentages right now. If you had to calculate a percent, you would just divide that number by 16. So like 1/16 as a decimal would be big T, big R, okay? Let's talk phenotype, okay? All right, so phenotype. Your possible phenotypes are determined by what your parents contributed, okay? You got to remember what the T's and R's represent. So you could have a phenotype that has big T, big R, which is tall with red flowers, okay? We'll make a blank for that and colon. Then you could have a phenotype that is tall with white flowers, which is big T, little r, okay? And then you could have one that is little t, big R, which is basically going to be short with red flowers. And then you could have the last one, which is little t, little r, okay? Now, the easiest thing for you to do to figure out your phenotype is to make sure you did your genotype right to begin with. If you did your genotype right to begin with, then you can use your genotype to figure out your phenotype. If you did it wrong, then you're going to be sorting through all those boxes, and you're going to be looking for how many of the boxes have a big T and a big R in it. How many boxes have a big T and a little r in it, you know, and so on. I'd rather use the genotype from above. Here's how you use it. So I'm going to go through my genotype, and I'm going to look for big T's and big R's. So this guy has a big T, big R. This guy has a big T, big R. This guy has a big T and a big R in it. And that guy has a big T and a big R in it. Does this one have a big T and a big R? Nope. So you can't count that one. You stop right there. So if you did this right, now all you got to do is add up these numbers. 4 plus 2 plus 2 plus 1. Those all have big T, big R. Last I checked, 4 plus 2 plus 2 plus 1 is 9. So we put a 9 right there. Okay? Next, I'm going to go through and look for big T, little r. So here's a big T, little r. Here's a big T, little r. Does this guy have a big T, little r? Nope. Can't count them. Got to stop. 2 plus 1 is 3. Okay? Then I'm going to go little t, big R. So there's a little t, big R. Here's a little t, big R. Does the last one have a little t, big R? Can't count it. So 2 plus 1 is 3, and you're good to go. With 1 left over, that goes there. So your phenotype ratio is a 9 to 3 to 3 to 1. All right, ladies and gents, if you can pull off that dihybrid cross, there is literally nothing I can throw at you that you can be fooled by. All right. So this video's covered how to do a monohybrid cross, the critical vocab. I've shown you the steps. I hope you've learned lots. Keep practicing at home, and I'll see you next time.